Bonjour. First of all, I want to thank a lot the organizer of this IOP Virtual Congress. In this symposium, we will address the initial brain development in premature newborns, in fetuses and infants from both a structural and a functional point of view. First, I'm very happy to welcome Dr. Jessica Dubois. She has a PhD in neuroscience. She is Director of Research at Neurospin in France. She will provide case structural information about the developmental brain process in typical preterm infants, in longitudinal studies, and using different aspects of MRI. She will talk about microstructure and maturation property of gray and white matter. This is really capital. We all rely on such beautiful structural data, and it's definitely important to better understand the emergence of cognitive functions. I'm also very happy to welcome Professor Hubert Press. He has a PhD in physics and neuroscience. He is the director of the FMEG-Center at the University of Tübingen. He will talk about learning hierarchical rules in fetuses and in newborns using local global auditory paradigms with fetal and neonatal MEG. And you should know that he is the only one in Europe using the fetal MEG. This is important to address how fetuses and newborns process input from the environment. I also want to welcome Professor SARA Morimi. She has a PhD in neuroscience. She is professor at Ferdowsi University in Mashhad in Iran, and she is researcher in Université Picardie at Amiens in France. She will talk about rhythm perception in premature neonates. She will address the question whether premature use the predictive model and develop top-down prediction about upcoming auditory events. We'll do that using HD-EEG with 128 electrodes in premature neonates. This is very interesting as it suggests that premature already develop high-level processing of environmental input. Finally, I'm very, very pleased to welcome Laurel Traynor. She has a PhD in experimental psychology. She is director of the McMaster Institute in Hamilton in Canada, and she will talk about the brain of 6 months old, how the brain of 6 months old infant extracts the beat and metrical structures in music using here also HD-EEG. This is critical for perceiving and learning language and music, which are key functions in social interactions. So definitely I'm happy to welcome everyone, and let's listen to each talk. Merci beaucoup, bonne journée.
Hello, everyone. So in this talk, we're going to talk about the exploration of the structural basis of early brain development with MRI. So you all know that the baby is acquiring many capacities after birth, and during childhood he's able to learn many cognitive capacities also, and all this relies on the intense development of its brain from birth on. And this growth of the brain is actually very intense during pregnancy and the first months after birth, which is quite a particularity of humans compared to other non-human primates. And this enables the environment to play a huge role in the development of the human brain. And any disturbances in these early processes might lead to long-term neurodevelopmental disorders. So understanding the early development of the brain is an important thing. So in this presentation, I would like to discuss with you 3 different things. First, how we can explore the structural development of the brain with MRI. Then we will focus on the auditory and language networks regarding given the topic of the symposium, and we will investigate some of the structural basis for the development of these networks. And then in a third part, I will try to give you some highlight of the functional correlates of this structural development using combined EEG and MRI studies. So let's start with basis of structural development. So the development of the brain is relying on many mechanisms which progression is quite complex during the pre and postnatal periods. All these mechanisms are actually overlapping in time, and here we will mainly focus on this time period, which is the last weeks of pregnancy and the first months after birth. So during this period, the connections are formed, they become mature, and so on. And we will see how we can explore this with MRI. So actually with MRI, we are exploring different mechanisms from the growth of the brain to the myelination of the white matter. How can we do that? We use these nice images that have been obtained in preterms in some infants and a child and an adult for comparison. And as you can see, the brain is changing a lot on these images, both on anatomical T1 weighted and T2 weighted images, but also on diffusion images and also on quantitative maps, such as the one shown here, which is quantitative T1 relaxometry. Based on these images, we can have insights on the growth and the folding of the brain. As you can see here, it's very intense after birth, and also the little movie shows you how the brain grows and folds during the last trimester of pregnancy. And we can quantify some parameters such as the brain size, the cortical surface, and the folding index. And all these mechanisms are actually increasing. Such parameters are actually increasing with age. We can also measure the microstructural development of the cortex. So you might know that connections are formed within the cortex, first with an overproduction, and in a second step you have a selective pruning of the connections just to keep the ones that are relevant given the external stimulation. And this process of growth and pruning is actually dependent on the function and on the region. And as you can see, it's earlier for the visuals than for prefrontal cortices, for example. So we can measure this with diffusion imaging mostly, as we did some time ago for preterm newborns that was scanned at 30 weeks of postmenstrual age and at time equivalent age. And we measured the parameter of diffusivity, which decreased with the maturation in the cortex. We could also use other parameters to do what we call multiparametric MRI, and we focused again on the cortical maturation in typical infants between 1 and 5 months of age, and we could show that the maturation was more advanced in some regions, in red here, compared with others, and these more mature regions were actually the primary sensorimotor regions, which were also highlighted through myelin staining by postmortem studies a century ago. Besides, the connections grow within the brain within the white matter, and again we can see first the development of projection bundles, then of commissural bundles, and then of associative bundles. And we can map these connections with diffusion MRI and tractography. At birth, actually, the long distance fibers are mostly in place, while after birth they become mature and the short distance connections are formed. These connections, they mature through the myelination process, which also take place at different rates across the regions, as Flechsig and Yakovlev shown with postmortem studies. And this process can last until the early adulthood. And again, with multiparametric MRI, we can show these differences in the maturation between tracts, and we could show that the primary sensorimotor pathways are maturing early on, while the associative pathways have a later and protracted maturation. So these are the structural basis of the developing brain. For the developing brain, now if we focus on the auditory and language networks, what are the specificities of this network? So First, you might know that it involves mostly regions, a network of perisylvian regions, which are regions located in the temporal, parietal, and frontal lobe. And studies by the group of Fabrice Valois and of Ghislaine Dehaene-Lamberts could show that these same regions are already involved very early on for the perception and processing of speech. So we focused on these specific perisylvian regions, and we could show with a diffusion MRI that the microstructural maturation is very intense during infancy. As you can see, this parameter of diffusivity is decreasing with age. And we also observe an important maturation in the white matter tracts that are involved. And here you have example for the acoustic radiations and the arcuate fasciculus, which is the big tract connecting the frontal, parietal, and temporal regions. Actually, in adults, people have described two pathways involved in the language network, what they call a dorsal pathway and what they call the ventral pathway. So we try to dissociate these pathways in the infant brain and to investigate their differential maturation. So to do so, we reconstructed a set of tracts in infants, and we evaluated their maturation, again with diffusion MRI. And we could show that the ventral pathways show an early maturation, which is actually in line with phylogenesis because this ventral pathway is predominant in non-human primates. Besides, the dorsal pathways show a delayed maturation, but also an intense maturation during infancy. And these pathways actually have been described to be involved in the multimodal integration of auditory, visual, and the motor aspects of speech. So this suggests that this multimodal integration is quite important in infants for them to learn to produce speech sounds in relation to the auditory and visual information they receive. So with the process of imitation, these dorsal pathways might be particularly involved. Besides, you probably know that there is an important functional lateralization of the language network, and it's mostly lateralized in the left hemisphere, which is already the case in infants as young as 2-3 months of age. So we investigated whether there is some structural basis for this functional lateralization. And first, we observed that it is actually the case in terms of growth and folding, and the perisylvian regions are the only ones which show these huge interhemispheric asymmetries at the level of the superior temporal sulcus, but also at the level of the Broca's region and the planum temporale. This is the case already in premature newborns, and it is also observed in infants, children, and adults. There are also some asymmetries in the microstructure of cortical regions in infants. This was first observed at the level of the superior temporal sulcus, based on an index of maturation with T2-weighted images. And we also observed this with diffusion MRI in the superior temporal gyrus. And both studies actually suggested a more mature superior temporal sulcus and gyrus on the right side. But this asymmetry might reverse during infancy, as in another study, we observed that the asymmetry is reversing with age from a right advanced maturation to a left one. There are also actually some interhemispheric asymmetries in the white matter. In infants, we observed a huge difference at the level of the arcuate fasciculus, as you can observe on the left side, and this is very similar to what have been described in adults. When we quantified the diffusion parameters in the different bundles of the language network, we observed that the arcuate fasciculus is the only tract which shows a huge asymmetry with a more developed arcuate fasciculus in the left hemisphere than the right. So I hope that I have shown you that we have strong asymmetries and already an early organization of the auditory and language network at the structural level, and this might support the functional development of this network. So that's what we will see in the third part of this talk. And first, I would like to describe a study that was performed with EEG and evoked potentials following the monowall presentation of stimuli, so the presentation only in one ear. Then we can observe some responses in the contralateral hemisphere and the ipsilateral hemisphere compared to the side of stimulation. In typical infants, when you measure the latency of these responses, you observe that the ipsilateral left responses are delayed compared to the other ones. But in the same study, we also studied infants with corpus callosum agenesis, and in these babies, we observed no difference in the different latencies. So this suggested that in typical infants, there might be a transfer of auditory information from the contralateral to ipsilateral hemisphere compared to the side of stimulation, and this transfer might be asymmetric in favor of the left hemispheres based on the callosal fibers. So this is something that still needs to be confirmed, but this transfer seems to be mediated by the callosal fibers connecting the two hemispheres. In typical infants, we also tried to compute these latencies and see how they change with age. So we measured these latencies both in a binaural paradigm, but also in a monaural paradigm when the stimuli were only presented on one side. And in each case, we observe that the latencies of the responses are decreasing with age. And this decrease in latency is supposed to rely on the myelination of the pathways that conduct the information. And that's what we aimed to investigate in another study. But before describing what we found for the auditory system, I would like to describe our results for the visual system, because the results are quite simpler to understand. So for the visual system, so it's a system that matures very early on following birth, and we observe that the latency of the first response, called P1, is decreasing a lot with age. And this is supposed to rely on the maturation and the myelination of the optic pathways, and among which the optic radiations. So in a study, we correlated the speed of response to visual stimuli to the myelination of the optic radiations that we evaluated with diffusion MRI. And we observed a strong relationship between the two. And actually, in this study, the visual stimuli were presented only in right hemifield one hemifield at a time, and then we could see a transfer of response from one hemisphere to the other, and we further correlated this speed of transfer to the maturation of the tract which transfers the information, which are the visual callosal fibers. And again, we could find a strong relationship between the speed of the transfer and the myelination of the visual callosal fibers. So for the visual system, we have a strong structural functional relationship. And the question is, do we see the same? Do we observe the same for the auditory system? So let's go back to our protocol for which we have this monaural presentation of stimulation and the contralateral and ipsilateral P2. And so the red circle is not at the good place, but we focused again on the left ipsilateral response, and we observed that this left ipsilateral response is actually its speed is actually related to the microstructure of some tracts, and the tract of interest we had was the auditory callosal fibers, because as we showed before, we observed that this response might be dependent on the transfer of information by callosal fibers from one hemisphere to the other in typical infants, but not in infants with callosal agenesis. So this study actually suggested an important role of interhemispheric connections in the development of lateralization. So I hope that I've shown you that we can combine different modalities of imaging, e.g. MRI, to investigate different aspects of brain development, both structural and functional aspects, and that the two are related, even if the relationships are harder, more complex in the auditory and language networks than what it can be for the visual network. So to conclude, the infant brain really shows an early architecture in networks, and it also shows an intense postnatal maturation with some plasticity and sensitive periods that are different between the functions. And what we can observe at the brain level is in line with the functional development and the behavioral acquisitions of the babies. So correlating the different observations is very important to better understand the development of babies. So for this, we need to conduct longitudinal studies combining several approaches, and this is really important to better understand typical development, but also for a better understanding of neurodevelopmental disorders that affect an important number of babies and children each year. So I thank you for your attention, and I would like to thank also all my colleagues from the Neonatal unit and the Neurospin Center. I would like also to thank the parents and the babies who participate to this research. And if you have any questions, I hope that you will join the live Q&A. Otherwise, you can send me an email. Thank you.
Dear organizers, first of all, let me thank you for giving us the opportunity to present our data in this interesting symposium. Dear colleagues, I want to talk today about magnetoencephalographic signatures of hierarchical rule learning in newborns and fetuses. This adds to our knowledge the first data on higher order cognitive processes recordable in fetuses. So how can we address the very early cognitive processes? And the way we are using it is by a device called SARA 2 SQUID array for reproductive assessment, which is installed at the University of Tübingen. And it is based on the magnetoencephalographic technique. It is a dedicated device to record fetal and newborn brain and heart signals. And you see it here in the upright position and here in the supine position. And you see here's his shell, which is shaped after the maternal pregnant abdomen. And it includes 156 sensors, so-called superconducting quantum interference devices, which are capable to record magnetic signals. So in general, the idea of the recording is that we record signals generated in the fetal body by electrical currents, and we measure the corresponding magnetic fields by the sensors covering the abdomen. So in general, a pregnant mother is laying over this sensor array, and we can record signals either in the spontaneous mode or also due to stimulation. And what you see here is an air-filled balloon, which we can use for delivering auditory stimuli, tones, sounds, whatever you like. But we also have the opportunity to also measure light and event-related responses. In addition, we can attach to the device a cradle and position a newborn over the sensor array and record magnetic signals also from the newborn brain. There are a lot of challenges in this kind of recordings, and I just want to give you a glimpse on the ideas behind it. when we record fetal brain signals, the largest signal is the maternal heart, and we have to attenuate the maternal heart signals, which is shown stepwise on the left side. So signals are rather strong of the maternal heart, and if we average, you see a clear maternal heart signal, which shows a specific topographic distribution over the sensor array. We can attenuate, and the leftover is mainly the fetal heart, which, for example, shows a dipolar structure, and this can be also attenuated. And the leftover we can record as standard ways we are performed in EEG or MEG recordings, where we can, for example, perform a trial averaging for the analysis of event-related fields, and, for example, we can extract the latency of event-related responses. So regarding the hierarchical rule learning, it has to be taken into account which processes are already established in the fetal life of humans. And we and others have shown that auditory event-related responses can be recorded after 24th week of gestation, and that the latency decreases over gestation. In addition, we showed that fetuses show mismatch-like responses to auditory stimulation in the last trimester of gestation, indicating that basic memory processing are already established in the fetal period. In addition, we have shown that the fetal behavioral state, which can be classified similar to newborn behavioral states, influences the event-related response latency. So just to give you an idea, I show you here the responses of fetuses on the left side and newborn on the right side to standard and deviant tones. And you also see the subtracted waveforms indicated the mismatch response. And you see certainly the responses are different between fetuses and newborns, but you also see in the case of fetuses a mismatch-like response in the range of 400 milliseconds, which is similar to the newborn mismatch response. So if we want to address hierarchical rule learning, we have to think about, okay, what is a suitable paradigm to use in this context? And what we selected for this approach is the local global oddball paradigm. And the general idea of this paradigm is that a participant has to maintain a global rule in contrast to a local rule in memory, and general assumption is that this requires some kind of conscious processing. This paradigm was used as a correlate of consciousness in different scenarios, and the global mismatch response is taken as an indicator for the ability to maintain the global rule. So why is it interesting for us to use this paradigm? Because there was an initial study by Basira et al. In 2014 who showed that newborns are capable to form a memory trace for the global rule at the age of 3 months. And so we wanted to address the question, okay, is it already visible in newborns and even in fetuses? So just to give you a little bit of background to make it more understandable, so the hierarchical rule learning, the local global paradigm, we have a sensory input, and on a first order hierarchy, we have a local rule, and on a second order, we have a global rule, which is integrated over the local rules. And what we used in our paradigm is an auditory local global paradigm, which consists of 2 different tones. One is called standard tone, which was in our case 500 Hertz, and a deviant tone 750 Hertz. And the study itself is distributed over 2 phases. One is the learning phase, where the local rule is learned, and a testing phase. And what you see, we use 2 different blocks. And for example, block 1, the local rule consists just of a sequence of 4 standard tones. And in the testing phase, this learned sequence is presented in 75% of the trials, and in 25 of the trials, it's replaced by a sequence of 4 tones, where the 4th tone is a deviant, and this is in one case a global deviant, but also a local deviant. In the second block, we used a different rule, where we have the local rule is a sequence of 3 standard tones with followed by the deviant, and this is presented in 75% of the trials, and in 25% the local deviant is in this case just a sequence of the 4 standard tones. So the tone duration was 200 milliseconds, the intertone interval 400 milliseconds, the sequence duration of the 4 tones were 2 seconds, and the intersequence interval 1700 milliseconds. So the first question, and this is work mainly performed by Julia Moser, a previous PhD student of my lab, is are newborns in the first weeks of life able to process complex auditory input containing first and second order regularities? And if yes, what is the impact of the infant behavioral state? And the study population were 33 infants, and 20 of these infants had at least 1 complete block with sufficient measurement quality. 17 of these recordings were block 1, 18 block 2, and in 15 both blocks were recorded. The age of the newborns was between 13 and 59 days, and there were 12 female and 8 male. First of all, let me present the data on the local rule, and what you see on the left side in gray are the response is the magnetic field strength to the local standards and the local deviants. The local standards are in gray and the local deviants in green, and what you see is that the magnetic field response is increased to the local deviants. In addition, if we compare the different conditions, as you have seen in the block design, you see that in this early mismatch window between 200 and 400 milliseconds, there is mainly a difference of the local and global deviant to the local and global standards. And if we take a further look into the data, what we see is if you look at the deviant and the global deviants and the global standards, and we take a look over the sequence, we see a significant difference in a very early time window and in a late time window, which is here depicted in gray. And for what you see on the right side is magnetic field strength in Tesla for the early and the late time window, and you see that the global deviants show an increased activation compared to the global standards. But we also have to take into account the behavioral state, and how can we address the behavioral state of the newborns. A rather pragmatic point of view is to use the newborn heart rate variability, and what you see on the left side is the distribution of the heart rate variability for the 15 included subjects, and what we took for this analysis is the extreme, so the newborns with the lowest and the highest heart rate variability. On the right side, you clearly see that the response to the first tone of the sequence is clearly different between the subjects with a low and a high heart rate variability. If we took a closer look in the global rule response in the early time window, which was shown before, we see that again, as indicated before, the general deviant, the local and global deviants, SSSD condition, showed a significant difference between the other conditions, and we have not seen any effect of state. If we look at the late time window between 720 and 745 milliseconds, we clearly see that the global deviants elicit a higher response in the high heart rate variability group compared to the low heart rate variability group. And this is also shown on the right side, where the different conditions are shown in a single case. And you clearly see that the global deviants are showing the higher activity. So this indicates that newborns show signs for learning of first and second order regularities, and that the second order rule learning is modulated by behavioral state. If we move on the fetal case, the question is the fetal brain in the last trimester of pregnancy able to process complex auditory input containing first and second order regularities? And if yes, how does this ability develop over the course of gestation? Again, we had a study population of 60 participants. 56 we were included because we had to interrupt some recordings. The number of data sets were 200 from 105 recording sessions, and 122 data sets showed us what we had as a criteria of valid brain localization. And we had 41 with brain localization in both recordings blocks, and the gestational age range was between 25 and 40 weeks, and the mean was 32 weeks and 32 weeks roughly. So the number of subjects included were 43, and 19 of them were measured several times longitudinally, and single participation was 24, and the fetal sex was nearly equally distributed between 19 female and 24 males. So what I wanted to show you at the beginning, we developed several tools adapted to this task, and what you see here is a cluster extracted showing the highest brain activity, and what you see is in blue the indication of the start of each of the tones in the sequence, and in red the average, the root mean square over these different tones. So for this analysis, we focused on the response between the difference between T4, tone 4 minus T3. And what we have seen is that there is a large difference in the late response window between 350 and 700 milliseconds, which showed again that the global deviance elicited a higher activity compared to the global standards. To further work on this, we also again looked at the vigilance measured by HRV in the fetuses. And as I told you in the beginning, there are certain parameters which can differentiate between behavioral state of the fetus. And we took into account again, like in the newborn, the age, the heart rate variability. And this is a parameter which can also differentiate between quiet sleep and active sleep. And again, we took into account the difference. And what we have seen is that the high heart rate variability showed this difference, and the low heart rate variability did not show the difference between the global deviance and the global standards. In addition, we looked into the developmental aspect of this global rule response, and this separated the group in before 34 weeks and after 34 weeks. And what we have found in this study is also that the global rule response develops rather late at the end of the gestational age, starting around 35 weeks. So taken together, we have shown that fetuses show signs for learning of second order regularities. We have seen a rule learning ability is modulated by behavioral state. The ability develops over the course of gestation and indication, and both the newborn and the fetal data that indicates that conscious processing is established in humans rather early, even in the fetal period. So thank you very much. And at the end, I want to thank my group in Tübingen and also my collaboration partners. Thank you very much.
First of all, I would like to thank the organizers of the Congress. Today, I will talk about our project where we investigate rhythm processing very early in the course of development, and particularly I will present our latest results on rhythm perception in premature neonates and the underlying mechanisms. Well, first of all, what is rhythm? Rhythm refers to the relative temporal relationship of auditory events. Temporal regularities between the onset intervals of events create rhythm that is perceived against symmetrical background. These temporal regularities are present at multiple hierarchical levels in both music and speech. Of course, it is more salient in music compared to speech. You see, for instance, the rhythm tree drawn to show the temporal relationships over the first measure on the image on the right. But why is rhythm important? Temporal grouping of auditory events and perception of rhythmic patterns is central to how we recognize temporal structures in sound sequences. The temporal regularities, or in other words, the rhythm, enable predictions about upcoming sounds. This predictive factor is important for music and speech perception, as well as motion creation and adaptation. Rhythm perception has been addressed in the context of predictive coding. In recent years, predictive coding has provided a unifying theory of the neural mechanisms underlying predictive processes in auditory comprehension. In this framework, tone pattern learning and prediction encompass a process of optimizing an internal neural model of temporal regularities. This is done through reciprocal bottom-up and top-down neural circuits. At the cortical level, this involves the primary auditory cortex, auditory association cortex, frontal and motor cortices. When predictions fail, the brain generates prediction error signals which are processed through neural hierarchies to revise the model's predictions. I will not go into further details, but I invite you to read the references provided on the bottom right of the slide for further explanations and details. Rhythm processing begins very early in development. We are exposed to rhythmic patterns from the very early stages of prenatal development. In fact, as early as 25 weeks gestational age, the fetus hears the isochronous rhythm of the maternal heartbeat and respiration. He is also constantly exposed to the environmental sounds originating from outside the uterus. Despite the attenuation of sound and distortion of its frequency content by the maternal tissue, the rhythmic information reaches the fetus. However, the infants born prematurely, especially the extremely premature neonates, are deprived of this rhythmic sound experience, and instead they are exposed to the extrauterine environment, the NICU. Unfortunately, this could lead to suboptimal auditory development. In fact, we know that some developmental disorders related to language and communication are associated with timing and rhythm processing deficits. Well, considering the importance of rhythm, the question is, can we recognize the deficits in rhythm processing early in the course of development and then use it as a neurobiomarker? To go into this direction, we aim to characterize rhythm perception in the last trimester of gestation, especially in preterm neonates and then infants born prematurely. As we discussed, auditory rhythm processing might begin from the very earliest stages of development. However, it has to be taken into consideration that the underlying neural circuits and mechanisms that process such information are undergoing rapid and dynamic evolution. At our period of interest, neural migration, dendrite differentiation, as well as synaptogenesis are happening. at approximately 28 weeks gestational age, thalamic afferents bathing in the subplate are relocated to the layer 4 of the cortex, shown in red on the right side of the slide. This period is also marked by the rapid emergence of short-range connectivity in addition to the long-range association pathways. While considering all this information, we pose the first question. Does the premature brain at birth, in fact, is the premature brain at birth capable of detecting rhythm or detecting the temporal regularities in a rhythmic pattern? To answer this question, we go into the NICU and record the neural activity of the premature neonates shortly after birth. This is done while they are being exposed to different types of auditory stimulation. Well, the stimulation protocol that I present today was specifically designed to address the neural response to rhythm deviations. For this purpose, we used a simple rhythm, a rhythm in a 2/4 meter. As you see in the image, we had for the standard condition 1 long tone followed by 2 short tones. And in the context of an oddball paradigm, this stimulation, this standard condition, was repeated. And then in the middle of the standard conditions, we had a deviant rhythm condition, in which the last tone, as you see on the image, the last tone arrived earlier than expected. And this created a deviation from the standard rhythmic pattern of the stimulation. We stimulated the preterm neonates with this protocol in the context of an oddball paradigm, and the age of the premature neonates was between 30 and 34 weeks gestational age, and they were all tested 1 week after birth. While they were exposed to this auditory sequence, we recorded the neural response using high-resolution EEG. This protocol was presented to 19 healthy premature neonates, and in the next slide we will discuss the neural response of these 19 premature neonates to the paradigm. Well, again, here you have the stimulation protocol. We see the train of the standard conditions, in the middle of which we have the deviant trial, which is the last tone arriving sooner than expected. What you see here is the ERP response tuned to the onset of the deviant condition, to the onset of the last tone, the onset of the last tone. And what you see here are 3 curves. We have the deviant response, or the response to the deviant condition, the response to the control condition. The control condition was created by only the deviant tones presented constantly, repeatedly, without the standard condition, because what we wanted to measure was the response to the violation. And instead of contrasting this response to the standard condition, where the timing of the stimuli did not match, we contrasted the deviant condition to the control condition, which was created by repetitive presentation of the deviant condition. So what you see here in red is the neural response to the deviant condition. We have the neural response to the control condition, and in green we have the difference between the 2 curves. As we see, approximately between 200 and 300 milliseconds after the onset of the deviant condition, we see a deflection which is significant between the 2 deviant and control conditions, which is the mismatch response. So the brain detects the deviation from the standard rhythmic pattern by creating a response. This response has a positive polarity And we see that it results in a cluster over the frontal electrodes. You see the carto on the right of the image for the 200-300 millisecond window, and this response was followed by another deflection with a different polarity, negative polarity, which was significant again over the frontal electrodes with a pattern which was a bit different. So the mismatch response, we would say, consisted of two components, an early component followed by a later component of a different polarity. So what we see is that the premature brain creates a mismatch response to deviation from at least a simple regular rhythmic structure. So having the mismatch response in premature neonates, we then moved to the second question. The question is this rhythm processing in premature neonates a low-level processing limited to the primary auditory cortex, or is it like it is in adults, a process that involves a hierarchy of cortical processes with bottom-up and top-down dialogues, as we discussed in the context of predictive coding? Why did we ask these questions? In the previous experiences conducted in our lab, the activation over the frontal regions of the cortex was recorded during the neural response to deviations from the standard phonemes, the frequency contents of the stimulation. Having the activation over the frontal cortex provide the questions regarding the role of the frontal cortex in processing the novelty and its role during the mismatch response. In addition, we know that the ventral pathway connecting the ventral portion of the inferior frontal gyrus to the temporal cortex is present at birth, at least in newborns, despite the fact that the dorsal pathway is less developed. So having the ventral pathway developed in the newborns, and also the neural response over the frontal cortex in premature neonates, make us wonder whether the frontal cortex is involved in processing the deviation from rhythmic structures as well. And if this is the case, how is it different from what is happening in adults? Well, to make a parallel in adults, recently we have analyzed the mismatch negativity, the neural response to rhythm deviations. And in the context of predictive coding, we have studied the oscillatory activity over the frontal cortex and the possible role of the frontal cortex in processing the rhythm deviations. Using dynamic causal modeling, the mismatch response to different types of deviations, including frequency deviations, during deviation, or omission of a tone, has been studied. It has been shown that processing of the error signal, the deviation from the standard, is carried out through the hierarchy of different cortical structures, including the primary auditory cortex, the superior temporal gyrus, inferior frontal gyrus in the ventral pathway, with both forward and backward connections. Sometimes it's lateralized to the right, as done by Garrido. Sometimes it's bilateral with the involvement of the inferior frontal cortex, the bilateral inferior frontal cortex. So, going back to the premature neonates, we know that we have the mismatch response to rhythm deviation. And we know that this process in adults involves the frontal cortex in the hierarchy that I just explained. So the next question that we have posed is, like I said, is it a simple low-level processing, or as in adults it involves a hierarchy of cortical processing? To answer this question, we have to address the effective connectivity underlying the mismatch response shown over the ERP. In fact, it's the first window that we show over the ERP. So what we did, we tried to address the neural effective connectivity underlying this response shown on the right. To do this, we used dynamic causal modeling. We used dynamic causal modeling to evaluate our hypothesis about the hierarchical mechanisms underlying the mismatch response in premature neonates, and also to investigate the effective connectivity between temporal and frontal cortical sources. DCM, or dynamic causal modeling, makes inferences about the underlying mechanisms of ERP components and the corresponding changes in the coupling between equivalent current dipole sources using biophysically constrained neural mass modeling. It considers a group of specific models to provide evidence in favor of one model relative to others using Bayesian model selection. I will not go through the details of dynamic causal modeling. I've put the link to the software. We used SPM 12 for the analysis, and I've provided the references for those who are interested to go into the details of dynamic causal modeling. Well, to test our hypothesis, we created a pool of models with connections between different cortical areas. Notably, we have A1, primary auditory cortex, STG, superior temporal gyrus, and IFG, inferior frontal gyrus. In the models presented on the right, we have different types of connections. We have the inputs to the auditory cortex, primary auditory cortex, only with the stimulus. Then we have intrinsic connections, only for the primary auditory cortex. And then we have forward connections that move along the hierarchy, and then we have backward connections. We have different candidates in the model. In the model pool, we have models only with the involvement of the primary auditory cortex. Then we have models with connections to the superior temporal gyrus, and then we have the models where the IFG is involved, but only with forward connections. As you see in model family 5 to 10, IFG is connected either on the right or on the left or bilateral, but only with forward connections. And then we have another family of models 11 to 16, where IFG is involved, but we have both forward and backward connections. So the more complex model family is the third family, 11 to 16, where we have the involvement of the frontal area with both forward and backward connections. Well, here we see the results of dynamic causal modeling. Again, I want to say that this analysis was only done over a window shown in red on the right side of the slide, meaning the window from the onset of the deviant tone until the end of the first component of the mismatch response. In comparing the models also presented in this slide, the 16 models, we initially used Bayesian model selection. This was done to identify the model that best explained the mismatch response. In this analysis, the model circled in red, consisting of the bilateral primary auditory cortices, bilateral STG, and right IFG with intrinsic connections in the bilateral primary auditory cortex, showed the highest evidence. The connections in this model, the connections between the bilateral A1 and bilateral STG, as well as the right STG and right IFG, are bidirectional and include both forward and backward connections. So this is the primary results, and then in order to evaluate again the importance of the backward or top-down connections, in other words, between IFG and STG sources, we used post-hoc family-level inference. The analysis presented on the right side of the slide shows that the family of models with both forward and backward connections, meaning model 11 to model 16, showed stronger evidence compared to the model family with only forward connections, and also to the family where we had connections only between primary auditory cortex and STG. This suggests that rhythm deviant mismatch response in the current study requires the backward connections between the IFG and STG. In fact, to conclude, we can say that processing of rhythm deviation in the premature brain is not limited to the primary auditory cortex, and, as in the case of adults, encompass a hierarchy of temporofrontal cortical structures in a both bottom-up and top-down stream. There are limitations to our study. DCM is based on local interactions between excitatory and inhibitory connections, as well as long-distance excitatory connections. We know that the neural network dynamics as well as the local excitatory-inhibitory interactions are not exactly the same between preterm neonates and adults. However, in light of the evidence from post-mortem studies, we have assumed that the simplified local and distant interactions in DCM can be assumed for late preterm infants. Nevertheless, it has to be mentioned that this extrapolation has to be treated with caution. The results presented here are all done by Mohammad Reza Adalati, a PhD student at our lab who will graduate soon. I would like to thank my colleagues Mahdi Mahmoudzadeh and Fabrice Valois for the pleasure of working together and all the nice discussions and the suggestions. And finally, I would like to thank Pauline and Laurence for the two research technicians in the laboratory who did all the recordings presented in this study. Finally, I thank you for your attention.
I would like to thank the organizers of this interesting conference, and specifically the organizers of this symposium. So in this presentation, I'm going to talk about measuring infants' responses to auditory rhythms using EEG. Rhythms are fundamental organizing principles of perception, action, and social interaction. Rhythms are ubiquitous in biological systems. Rhythms are essential for locomotion, speech, music, and social interaction. And many species perform rhythmic movement for locomotion. Even this jellyfish with very few neurons can execute exquisite rhythmic movement. And here is more evidence that it does not take many neurons to perform rhythmic movement. And human communication systems, speech and music, are rhythmically organized, and rhythms are essential for social communication and social interaction. So why are rhythms so powerful? Well, here are three basic reasons. In perception, signals like speech and music unfold rapidly over time, and we need to organize or parse them into meaningful chunks or phrases in real time in order to decipher their messages. So underlying beat structures provide a scaffold for doing this. The regularities of rhythms enable us to predict the expected timing of important upcoming information, which in turn enables us to direct our attention to key points in time where important information is likely to occur. The importance of timing is clear in that all of the major developmental disorders are associated with timing deficits, including dyslexia, attention disorders, autism, stuttering, and developmental coordination disorder. Rhythms have several layers of structure. The most obvious is the rhythm surface. So shown here in red is a rhythm pattern, and when repeated, it sounds like this. So the notes are occurring at irregularly spaced intervals. However, an underlying 6-beat regular pattern, shown in black, or a beat, can be felt when we hear this rhythm. So notice that we generate this beat level internally. Our brain is looking for regularities and timing patterns, and we can easily figure out where the beat is and tap our foot or nod our head or dance to the beat. And we can feel beats even when there's no sound at that time. So our brains not only extract one basic regular beat level, but we group beats together to create metrical hierarchies of beat levels. So we can accent, for example, every second beat, like this, to form a march, 1, 2, 1, 2, 1, 2, or we can accent every third beat to form a waltz, like this, 1, 2, 3, 1, 2, 3. So in my lab, we have been asking whether the infant brain automatically tracks both the beat and meter levels in rhythm patterns, as has been shown for adults. So on this slide, I'll describe the stimulus we used in our initial study. We presented infants with the repeating rhythm patterns shown here, the same one I just showed you on the last slide, while we measured their EEG. And you can see here in panel A that the basic rhythm pattern is 6 beats long and takes 2 seconds. If we analyze the frequencies that are present in the stimulus, we see that the beat frequency in blue is 3.33 hertz, because there are 6 beats every 2 seconds. The duple or march frequency in red represents every other beat, so its frequency is half of that, or 1.67 hertz. And the triple or waltz frequency in green represents every third beat, so its frequency is 1.11 hertz. Now in panel B, you can see a Fourier analysis of this pattern, this stimulus, and indeed the beat, the duple, and the triple frequencies are all prominent. And of course, there are also some subharmonics and harmonics present. So we brought 6-month-olds into the lab. We measured their head while the parent filled out the forms and questionnaires. We distract them with Big Bird while we put on the EEG net. We adjust it, check the impedances, and we're ready to go. So infants listened passively to the stimulus while they watched a silent infant video to keep them still. So the first study was led by Laura Sorelli when she was a graduate student in my lab. I'm sorry, the tempo was slightly different here than in our later studies. So in this particular stimulus, the beat frequency is 3 hertz, the duple is 1.5, and the triple is 1 hertz. So here's a Fourier analysis of the stimulus that shows the beat, the duple, and the triple frequencies. And here is the Fourier analysis of infants' EEG while they listened passively to this stimulus. And you can see that infants' brains are tracking the frequencies in the stimulus very well. So here's the beat frequency. Here is the duple march meter frequency. And here is the triple waltz meter frequency. The beat frequency is actually enhanced in the infant's EEG compared to in the stimulus. Now remember that infants were simply listening passively here. So this tells us that infants' brains are continually tracking the rhythms of the auditory stimuli in their environment. In this study, we also examined effects of whether infants and their parents were attending music classes for babies, shown in purple bars, or not attending such classes, shown in green bars. What you can see is that there was no difference in how well their brains were tracking the basic beat frequency, shown here. However, those in the music classes were better at tracking the meter frequencies, both the duple meter, shown here, and the triple meter. This is correlational data, but it suggests that early experience particularly affects the processing of meter or grouping structure. So next we asked whether infants can be primed to hear this ambiguous rhythm in duple versus triple meter. In other words, can they maintain through top-down processing an internally imagined metrical interpretation? We know that adults derive their conscious interpretation of whether they perceive this rhythm as in duple or in triple meter, in part through top-down processes. So here's the basic idea. Through bottom-up processing, the sound is processed in the ear and through subcortical pathways to auditory areas. And the question is whether there is also top-down processing that influences whether the infant perceives either a duple pattern as shown here or a triple pattern as shown here. Now the accents are not actually in the stimulus that we present while we're recording the EEG. I've just put them here to indicate how infants might be perceiving the stimulus. So to examine top-down processing, we are looking for neural responses that cannot be explained simply by the sound input. So here is how we tested this using a priming method. So this study was led by my graduate student, Erica Flayton. Infants were presented with a priming stimulus that had intensity accents either in duple meter on the left or triple meter on the right. The priming pattern repeated 4 times. Then they received 16 repetitions of the pattern with no accents, so they heard the same test stimulus regardless of whether they were in the duple or triple priming condition. So during the 16 test trials, 75% were the standard stimulus. However, in addition, on 12.5% of trials, the note on the 4th beat was raised in pitch by a semitone, and on the remaining 12.5% of trials, the pitch of the note on the 5th beat was raised by a semitone. So note that the 4th beat is strong or accented in triple meter, but weak or unaccented in duple meter, whereas the 5th beat is strong or accented in duple meter, but it's weak or unaccented in triple meter. So the whole sequence of 4 primes and 16 test trials repeated 9 times per block, and we attempted to get infants through 3 blocks. So we measured EEG and examined infants' mismatch responses to occasional pitch changes. So MMR, or these mismatch responses, manifest as a positive peak between 150 and 250 milliseconds after onset of these occasional unexpected or deviant pitches in the sequence of expected or standard pitches. Now there's some evidence that it can be modulated by top-down processes in infancy. So for example, there is larger MMN for deviance on metrically strong than metrically weak positions. So here are our main results. These are difference waves, so they're deviant minus standard for frontal left electrode sites. Time is on the x-axis and amplitude is on the y-axis with positive up. So in the upper graph, you can see that MMR is larger for beat 5 than beat 4 when primed in duple meter. And on the lower graph, you can see that MMR is larger for beat 4 than beat 5 when primed with triple meter. So this tells us that infants show greater sensitivity to pitch deviance on beats that were perceptually strong according to their priming condition. So we conclude that infants can maintain an imagined or internally generated metrical interpretation through top-down processes. There was another interesting result when we examined frontal right responses. And here, regardless of whether the deviant was on beat 4 or on beat 5, MMR responses were larger in the duple group, shown on the top graph, than in the triple group, shown on the bottom graph. And this is consistent with the idea that it was overall easier to process the stimulus in duple than triple meter interpretation, Now, given that Western music is predominantly in duple meter, this might reflect early enculturation to the meters of Western music. Finally, when we compared MMR responses in those with one or more musical parents, shown in the solid bars, to those without a musical parent, shown in the hatch bars, those with a musical parent showed larger MMM responses in general at frontal left sites. So this result could reflect genetic or environmental effects, or some combination of the two. We can't tell from this study, but it's very interesting that the relation between infant and parent musicality is present very early in development. So to conclude, infants' brains automatically track the beat and meter frequencies present in a rhythmic stimulus. Infants can be primed to maintain a particular metrical interpretation of an ambiguous rhythm. And the data is consistent with early enculturation to the rhythms of your environment and early correlations between infant and parent musicality. So the brain is set up to track temporal structure and to maintain top-down metrical interpretations, even in infancy. So thank you to all the people in my lab, and thank you to the funders of this research, and thank you for listening.